Great. Thank you very much, Afric, and thank you very much for the invitation to speak at this conference. So again, as Susanna has mentioned, this is a follow-up on the study that was previously just discussed. Okay. So why would we focus on the microbiome with vitamin D? So as Susanna has previously mentioned, there is now a clear, distinct link between vitamin D supplementation and the immune system. And we know that there's a clear interaction between the microbiome and immune system, particularly in the digestive tract, where there's a constant state of flux or interaction between immune cells and the actual microbiome that's there, be that the commensal population that are involved in the breakdown of nutrients, or also then with the interaction with potentially pathogenic bacteria. So I suppose the question then is this vitamin D supplementation that we've seen in the previous talk, is that actually then affecting the microbiome, either that through interactions with the immune system or having a direct effect on the microbiome itself? Okay, so just to show where these samples are collected on, so this is taken from Susanna's talk. So this is at the T9 time point where the samples from the microbiome were collected. And we can see at this time point that we have a clear divergence between our control and vitamin D supplemented calves in both the indoor and outdoor systems. So we would think if there is going to be changes in the microbiome, we have a clear phenotypic population to identify these changes. So from the 48 calves that were involved in the initial trial, we took samples from 24 of these calves. So again, we have our control samples indoors, our vitamin D supplemented calves indoors, our control fed calves outdoors, and our calves fed outdoors vitamin D outdoors also. So from these cows, we took 2 samples to get indicators of both influences of vitamin D on calf health, but also as proxies for the rumen and to see if there's any potential impacts on nutrient breakdown. So from these 2 sample types, so we had 24 samples for the oral and 24 for the fecal, DNA was extracted from these samples, and we sequenced the 16S V3 V4 region using the Illumina-MiSeq. We then used KIME bioinformatic packages to do the initial read clustering to produce an OTU table, which then allowed us to do a number of different measures looking at bacterial diversity, taxonomic composition, and then also allowed us to do differential abundance analysis between both our sample types and also the housing types. So we wanted to look at the effect of housing on the microbiome, but also then the effect of vitamin D supplementation. And also importantly, are we getting a differential response between the indoor and outdoor calves in relation to the vitamin D supplementation on the microbiome? So how did our different sample types compare? So you can see here that we have our fecal and oral samples. So while this is probably to be expected that you are going to see a difference in the microbiome between these 2 regions, it was still interesting to look at and see that we have identified quite a distinct difference in the fecal and oral microbiome samples, irrespective of our housing type and supplementation levels. So these differences were identified using multiple dimensional scaling and Bray-Curtis distance matrix. So again, as expected, then we treated these two groups as different sample types. So we had two samples for our oral and our fecal. So next thing we wanted to look at, is there a considerable interest in influence of housing type within these fecal and oral samples? So what we first identified that within in the fecal samples in particular, we are getting a distinct effect on the microbiome from these 2 different housing types. So we can see quite a tight clustering of the calves that were raised outdoors in comparison to the separation in the calves that were indoors, but still distinct from the cows outdoors. So again, suggesting that we're going to get a difference in response or difference in the microbiome in these two housing types. And then interestingly, while we saw this in the fecal microbiome, when we look at the oral samples, we're not getting a clear distinction between the two housing types in the oral sample. So again, this is a relatively novel way to look at the oral microbiome in calves. So we're just interested initially to see how our different experimental factors were influencing the oral microbiome. So now moving on to actually the effects of vitamin D. So what we have here is alpha diversity measures. So we have observed, Shannon, Simpson, and Fisher measures of alpha diversity. We have our indoor housed calves on the left, and we have our outdoor housed calves on the right. So what we can see from all of these measures of diversity is that calves housed outdoors had a greater diversity based on all of these measures of alpha diversity that we actually looked at in this experiment. So again, going back to what we saw on the previous slides, quite a distinct effect of housing on the fecal microbiome. When we look at the actual effects of vitamin D, we're not getting as large effects of vitamin D on the measures of alpha diversity, but we can see in the control in red compared to the green, we are getting slight increases in alpha diversity, particularly in Shannon, with again, the cows fed vitamin D having increase in alpha diversity. So increased diversity is generally associated with improvements in health of the studied animal. So now getting into more specific examples of what are the impacts directly on the microbiome of the animal. So again, here we're primarily just focusing on our vitamin D vitamin D supplementation is irrespective of housing type, and these are our fecal samples. So what we can see here is by supplementing with vitamin D, we're getting reductions in CF231, reductions in Clostridium, which is known to contain a number of both commensal and pathogenic bacteria, and increases in the genus Duria and Osteospira, which are both associated with the fermentation of carbohydrates. Suggests by supplementing vitamin D, we're possibly getting improvements in the breakdown of the diet. So again, very interesting changes. And it's interesting to note that Osteospira is the most abundant genre that was identified in this study. So it's not just minor changes that are being impacted by supplementing with vitamin D. We're actually getting quite large impacts on the microbiome. Now moving on to our oral samples. So the first thing again to note is there's quite a distinct difference between the genera that are identified in the fecal samples in comparison to the oral, but we're still getting large impacts again with the supplementation of vitamin D. So we can see increases in Lactobacillus, which is again associated with improvements in health, reductions in Clostridium and Fusobacterium, Moraxella, Mycoplasma, which are all known to contain pathogenic bacteria. And then again, we have a similar effect on Osteospira, but not as stark as we see in the fecal samples. We did have a significant reduction in Ruminococcus, which again is a little bit difficult to interpret how this is going to actually impact the animal, as this is one of the main genus involved in the breakdown of complex carbohydrates. So how that actually is having effect in the oral samples, it's a little bit difficult to interpret at this point. But finally, just one thing that we thought was particularly interesting in this is then when you analyze the interaction between our vitamin D supplementation and the actual housing type. And what's interesting about this is that we're getting a different response when you separate out the housing types. In particular for Clostridium, you can see in the calves that were housed indoor, a significant reduction in Clostridium following vitamin D supplementation, whereas outdoors we're having no difference between the two groups. Then the opposite is seen in Oscilliospira, where there's no difference in the indoor housed calves, whereas outdoors, when we supplement with vitamin D, we're getting a significant increase in Oscilliospira, which again is associated with the breakdown of complex carbohydrates. So an interaction of the supplementation of vitamin D with the diet and housing type. So just to conclude, there's clear evidence that vitamin D supplementation impacts the immune cell profiles from Susanna's talk, and now we also see from my talk from that it has an impact on the microbiome. So the impacts on the microbiome are influenced by housing status as well as vitamin D, with a differential response based on housing type of vitamin D. So the next question, what we want to look into, is kind of what is driving these changes in the microbiome. Is it vitamin D impacting the immune system, which is then impacting the microbiome, or is there more direct effects on the microbiome in comparison to the impacts on the immune system? Thank you very much again for giving me the opportunity to give you this presentation.